My name is Ross Macdonald. I'm the CEO of Cynata Therapeutics, a next-generation stem cell therapeutics company. We are a publicly traded company, so I remind you to look at our website for information and at our filings on the Australian Stock Exchange platform. Cynata's focus is the development of our proprietary Cymerus platform technology to expand and commercially scale cell therapy products to treat serious chronic diseases. In particular, our focus is mesenchymal stem cells, or MSCs, which in this forum certainly need no introduction. A few quick words about our company. We are a phase 2 ready technology company with a highly scalable proprietary platform for the manufacture of stem cell therapeutics. We are listed on the Australian Stock Exchange. Our technology originated from the University of Wisconsin here in the United States. We're at clinical stage, and we have a globally experienced board and management team. Our market capitalization at present is around $110 million US. Our largest shareholder is Fidelity, and our strategic partner, Fujifilm, also is a large shareholder with just under 8%. A quick summary of our technology. We have a globally scalable, globally applicable scalable technology, a licensing-driven business model, successful clinical trial results, which I'll expand more on in a moment, multiple near-term catalysts with some very exciting clinical programs about to get underway, and of course we're active in the very attractive regenerative medicine industry. As I said, mesenchymal stem cells need no introduction, and the excitement around their clinical potential is evidenced by the large number of clinical trials that are underway around the world, around 930 clinical studies in a wide variety of diseases. And that number represents a steady growth over the past 8 or 9 years or so, as indicated by that slide. The interest in the therapeutic potential of MSCs is, of course, dictated by their biology, and it has been shown in many, many clinical and preclinical studies that MSCs have the capacity to regenerate damaged and diseased tissue, a profound modulatory effect upon the immune system, and can enhance and accelerate the recovery of tissue from injury associated with disease or other tissue damage. This has led now to more excitement as MSC-based therapies are reaching the market after many years of development. And so in Japan, for example, we have a product that is now on the market for graft versus host disease and a product that's been approved in Europe for Crohn's fistula. And we hope in the coming year or two that there will be many more products to follow. The challenge, however, we face in commercializing all cell therapy products focuses on the process used to manufacture those products. And as we are dealing with a living drug, the isolation, purification, and expansion of sufficient quantities for commercial application represents a major challenge to the industrialization of cell-based therapies. Autologous systems, obviously on a bespoke basis, are fine, but if we're talking about an off-the-shelf allogeneic product, as we are with our product and many other MSC companies are also considering that, then we face a major impediment which is driven by the need to access the cells from a living donor. And the processes that are used so far to generate MSCs for therapeutic application, sourcing them from adipose tissue or bone marrow, for example, we feel, in our view, is a flawed business strategy and a flawed commercial model. The reliance upon multiple donors and massive expansion of purified MSCs is really not a solution to the commercialization of these very exciting therapies. So we took a completely different view. We recognize that MSCs have very exciting biology which can be harnessed therapeutically, but we felt there was a better way of achieving the manufacturing scale-up that's necessary for commercial development. Embryonic stem cells are one route as a starting material, but for many people that represents a major barrier due to their ethical controversies and the association with the destruction of embryos to source them. On the other hand, the discovery of induced pluripotent stem cells in the last decade provided an alternative that represents a starting material which can be differentiated under appropriate conditions to essentially any cell type in the body. Our approach was to take iPSCs and to differentiate them into MSCs. The capacity of iPSCs to infinitely renew themselves provided one of the most important characteristics, and that was the ability to make lots of them, tons of material, which would be necessary if we were to address disease targets such as cardiovascular disease or stroke or osteoarthritis globally. So that ability to make mass amount of material consistently and reproducibly was the hallmark of our strategy. And so we sourced a GMP line from Fujifilm CDI, or as it was back then, just CDI, and using our proprietary Chimeras process, we expand those iPSCs, and then ultimately, in the final step of the manufacturing process, again using our proprietary system, differentiate them into MSCs. Those MSCs have all of the expected properties of an MSC as defined by the ISCT criteria, and of course we all know that that is a very, very broad criteria, and there's a lot of debate in the industry about what exactly constitutes an MSC, but nonetheless the MSCs that we derive from the iPSC route have those characteristics. Ultimately, it is necessary to prove that the cells are useful therapeutically, which is what we set about doing, having established the manufacturing process and, of course, a very strong preclinical database. As I mentioned, the IPSC approach addresses that Achilles heel of manufacturing cell therapies, at least for MSCs. That is, to provide a scalable and consistent manufacturing solution, and also, as we believe, cells that are much more potent and require fewer doses or fewer cells or lesser frequency of doses to produce a clinical outcome. And this, of course, goes to a lot of the literature in this area around expansion of MSCs, and that is that massive expansion protocols ultimately lead to a loss of potency. In our case, the expansion is at the level of the iPSC, so what we end up with at the conclusion of our manufacturing process is a very fresh, robust MSC, which has properties very similar to freshly isolated MSCs from tissue sources. And we're the only company in the world that has the capability of manufacturing products using this technology. What is very exciting, and I'm sure you're all aware of the enthusiasm now that is being generated by companies such as BlueRock and Takeda and Fate Therapeutics, which are using iPSCs as a starting material for their particular favourite type of cell. So whilst Cynata was a voice in the wilderness early on, we're now being joined by some very, very significant players in this field who have recognised the opportunity that iPSCs represent as a consistent, robust and reliable starting material for cell therapy manufacture. Our technology has now demonstrated itself in a very successful phase 1 clinical study in graft versus host disease that has been licensed to Fujifilm in a transaction that we announced a couple of weeks ago. So Fujifilm is now our partner to develop this product for commercial application around the world in GVHD. We plan to commence phase 2s in osteoarthritis and in critical limb ischemia in the next few months. The osteoarthritis trial is substantially funded by the Australian government, and at around 450 patients will be one of the largest MSC trials conducted to date. And we'll finally address that question, do MSCs work in osteoarthritis? This will be a very well-powered study, as you would imagine, with that number of subjects. The CLI trial, we hope, also will get underway in the next few months. In other areas, we have very, very exciting preclinical data, as shown on the right-hand side of the slide here, in conditions such as asthma, in heart attack, in addressing the tissue damage which occurs after an infarction. We have a program underway in sepsis, some very exciting data in diabetic wounds and cytokine release syndrome, where our MSCs have been shown to modulate the effects that are associated with the delivery of immunotherapy products such as CAR-T, which produce that cytokine storm, which is so damaging to patients who react adversely to those products. And some data also in coronary artery disease. So a very nice showcase of preclinical data, which is now supported by our clinical trial data in graft versus host disease, enabling any one of those applications to move forward into the clinic. Our license-driven business strategy is dictated by our need to establish value-creating events for our shareholder base. We can do so by building our platform independently, which of course is the typical model here in the United States, where you've got a very robust finance and capital markets ecosystem that enables companies to do that. But in Australia, it's a somewhat more challenging capital markets environment, so building a fully integrated biotech company is somewhat more challenging. On the other hand, we could also look at strategic partnering or strategic exits, and in fact, Cynata presently is the subject of a takeover bid by Nippon Sumitomo. The strategy, however, that's proven successful for us so far is to license our technology to big pharma companies, and our transaction with Fujifilm, which, as I said, we completed a couple of weeks ago, is one such example. And as a case study, it's a global license for GVHD, results in multiple cash flow events for the company through upfront payments and milestones on success, and then finally a double-digit royalty on product sales. And that represents that very important validation that comes with the partnering with a pharmaceutical company that's got the expertise, the resources and capability to fully develop a product. Turning now to our clinical trial, and I won't go into the protocol in great detail given the time, but GVHD is a particularly devastating disease, and the standard of care involves corticosteroid therapy, which fails in a significant proportion of patients, leaving those patients with no therapeutic option. And the outcome of acute steroid-resistant GVHD is typically very poor, with patient deaths occurring within a year or two in typical studies around the world. Our trial involved patients who had failed corticosteroid therapy. Patients received 2 doses of our product, 1 million cells per kilogram, 7 days apart, or 2 million cells per kilogram, 7 days apart. The patients were investigated for safety and tolerability, primary endpoint, of course, for phase 1, and complete response, partial response, and overall survival at various time points following the completion of the study. The trial was completed last year, and we announced the results as that trial concluded. Very, very exciting outcome. These patients, as I mentioned, typically have a very, very poor outlook. But in over half of the patients, we saw a complete response. Patients were enrolled in the trial typically at around grade 2 or grade 3 GVHD, so very sick, and all or around half of those patients had a complete recovery of their symptoms, and nearly all of the patients had at least 1 grade improvement in their symptoms. We had 1 patient die of pneumonia during the course of the clinical study, which is not unusual in GVHD, and 1 patient withdrew from the study, so they were unable to follow up. Otherwise, that would have been an even more exciting set of numbers there. But certainly, overall survival at day 100 of greater than 87% was an outstanding outcome in a typically very, very poor outlook patient group. As I mentioned, we now have a clear pathway to commencing phase 2 programs, not only in GVHD in partnership with Fujifilm, but also in critical limb ischemia and in osteoarthritis. An announcement a few days ago around our osteo trial indicates that will get underway in the first calendar quarter of next year. And as I mentioned earlier, a very, very large and very important trial to demonstrate unequivocally whether MSCs are useful in addressing the very devastating health effects of osteoarthritis. Other programs will get underway pending upon partnership opportunities and availability of capital. We're well positioned in the regenerative medicine industry with graft versus host disease, a fairly small opportunity. It has orphan drug designation here in the United States, but certainly important to get us moving into some of the more economically attractive opportunities such as CLI and osteoarthritis, which of course is a very, very large market opportunity at more than $11 billion market value. Finally, of course, the M&A market in the cell therapy space has been quite vibrant, and as I mentioned before, we ourselves are the target of a non-binding conditional takeover bid by Nippon Sumitomo at the moment, and our board is working through the process associated with that. Finally, to sum up, Cynata provides Australian Stock Exchange exposure to the growing regenerative medicine industry. We have a next-generation, scalable, consistent manufacturing process for therapeutic MSC products. We have an attractive licensing-driven business model which has already proven to be successful in our partnership with Fujifilm. Very good results in our first clinical study, which we hope will be replicated as we move forward in other indications. Endorsement by Big Pharma, and then finally we continue our license-driven business strategy as we engage with other companies and to look to other strategic partnerships pending on the outcome of our discussions with Dai Nippon. I'd like to thank you very much for your attention, and hope you enjoy the rest of the conference. If we have a couple of minutes for questions, if there's anything burning, I'm happy to answer that. Otherwise, we'll see you afterwards.